Hello, good morning, good afternoon, good evening, good day to wherever and whenever you're joining. This is, and welcome to your first of your InvertebrateR or your InvertebrateR assignments. And the first of two videos that's going to walk you through those assignments. In this first one, we're going to talk about the context for the assignments. And the assignments are both of the InvertebrateR assignments are written as an explicit examination of diversity in deep sea thermal vents and at the deep sea biota in general. So we're going to talk a little bit today, just bring everybody up to speed, remind you, because maybe you haven't thought about deep sea thermal vents since thinking about things in 2700. But we'll talk about them. We'll talk about some of the theoretical underpinnings, the ecological underpinnings to the kind of distribution that these vents have across the world. So we'll talk about metapopulations. We'll talk about the biological features that would connect those, and that's dispersal. And so for most of these, it's the larval dispersal. And how can we infer that? There's a lot of different ways to infer that genetically, and we're going to do a tree-based one using phylogeny. And then we're going to overlay that with a context of deep sea mining and threat or potential development threat. Talk a little bit about the assignment, and then there's another half-hour video that you can click on later on that works through a parallel example of the R data set that you're going to be working with. So my suggestion is for each of these videos, this context and then the explicit example one, is they're divided up into sections. Watch them slowly. There's no need to race through these things. So think about the deep sea vents, think about the blah, 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 blah. And so pause, reflect, think about things, do some parallel reading, that kind of thing. And the same holds true for the parallel video in R. Pause that one a lot as you're going through, so you can kind of match the speed and the rapidity with which we're going through. All right, let's begin. So, deep sea vents, crazy places, gorgeous places. These concentrations of almost unimaginable dense life associated with almost unimaginably complicated physical conditions, high temperatures, extreme pressure, and no sunlight at all. So light in a place that up until relatively recently, we didn't think there would be any life at all. These are some pictures that date back to just under 50 years ago, to 1977. Our awareness of this habitat is comparatively recent, and it dates back to an expedition from Woods Hole Oceanographic Institute that Bob Ballard led. Bob Ballard has done a bunch of things. You probably, if you're a fan of the sitcom 30 Rock, there was Bob Ballard is a friend of Jack Donaghy. So Bob Ballard is kind of, as oceanographers, as marine scientists go, he's pretty famous. And this was some of the start of that fame. So he and 30 other researchers were cruising around off the South American coast in the vessel Nor. And you can see them here at their data, and here kind of priming this little bit of ingenuity, which they called Angus. And Angus was a submarine that was built to do two things. So this is pre-streaming, pre-whatever, right? Like, it's basically just you're jigging. Like, if you're a fisher, jigging, you just put the weighted hook over and you're kind of bouncing it up and down. And that's literally what they were doing, is bouncing it up and down and knowing that it was going to record temperature and that it would automatically take photographs. And so what they would do once they reeled Angus back in was look for hot spots in that time series and then develop, because they were looking for these thermal vents, and then they would develop the film associated with those hot spots. And what they found, so this is them, and so you've got kind of bouncing and bouncing and bouncing and bouncing, and then dense life. And this surprised them. So there you see, at a slightly later date, not in 77, but this is the same location off the Galapagos. Dense, dense, dense life. biology beyond the reach of the sun. So this immediate, incredible discoveries of a highly diverse community of organisms that were endemic and unique and hitherto unknown to science. So Pompeii worms, mollusks with shells made of metal, yeti crabs, incredible symbioses, bacterial symbioses that let them live, that permit life to occur in these places. And what we've learned since Bob Ballard was jigging with Angus off the Galapagos in 1977 was that these deep sea hydrothermal vents are distributed around the world. And you can see kind of a rough map of them and where we know. So these are the hot spots, are some of the places where, or the colored dots here, are some of the places where expeditions have actually explored. And that what we know from a geological perspective is that these are the rifts and the vents that the vents would track. So globally distributed. Now, when I think of biota that's globally distributed, I generally think, oh, that's a lot of, that's enormous, right? So probably not very, not exposed to the problems of small population size. But that first bullet, they're globally distributed, but they exist, the environment that it's really small, 50 square kilometers over the world, distributed in very thin intermittent strips. Okay, and so how big is 50? That's generally smaller than the total surface area of the city of Guelph, but stretched in a very thin line. But each of those tiny little pockets of life, those hydrothermal vents, is a site that's characterized by many, many unique species with a relative biomass that compares or rivals or exceeds coral reefs or tropical rainforests. And this density and diversity and isolated kind of pockets of life would be very threatened to a development such as mining. So let's take a look at this. Is an estimate of species richness for a particular lineage of mollusks. So we've got from one estimate of this group to 32, and you can see in these different areas, these vent communities around the world, that the diversity ranges. So light green or light color is low species and high is dark. And then we've got different biogeographic regions all over the world. So there's isolated populations with a global distribution. So it's patchy, highly endemic. How connected are they by dispersal? This is a question. And so one element that ecologists, that biologists would bring to the study of this kind of a patchy distribution is the concept of a metapopulation. So a metapopulation is something that you've probably bumped into before, but to bring us all up to the same kind of an understanding without going into any of the math of whether it's a core satellite 11 style or a patchy population, essentially with a metapopulation is Levins in 1969 made a really simple mathematical argument that you could convey kind of in a back of the napkin, a sketch on a napkin, like this kind of a thing, where a metapopulation is a series of populations where all the populations have some non-zero risk of extinction because, so these are extinct little habitat patches here. These are extant or population habitat patches where there are members of that species, that community living. And that non-zero risk is because these arrows connect. And so there is, the non-zero risk of extinction is intimately tied to the dispersal ability of the species or the species in question. And so populations are spatially structured into assemblages of local breeding populations, and dispersal between the populations allows recolonization. And so it was intuitively appealing to a lot of people because we tend to see, as humans, when we see the world, we have a bias towards kind of a binary, breaking things down as, well, that's a place, that's habitat, and that's not habitat. And so amphibian biologists do this all the time, where we look at a pond and say, that's a pond, that's not a pond, so that's not habitat, this is habitat here. And maybe the non-habitat interferes or accelerates or prevents dispersal. But it's intuitively appealing, finally, because it lets us think about populations across the landscape kind of in the way that we would think of individuals within a population. So it extends that kind of that thinking up. And so the data is relatively simple for a metapopulation on one level. If you put aside for a second measuring dispersal, what we've got here, flipping through, is a series of black and yellow shapes. And these are habitat patches for a particular species, in this case of, I apologize, this is an amphibian, but it's skipping through and it's showing these blinking lights turning on and off as populations here across this matrix. So different populations across rows, and then different years across columns, and so they would be colonized, and then this one would remain colonized. And this population here would be colonized and then go extinct. Colonized, extinct, colonized, extinct, colonized, extinct. And so this was intuitively appealing, this concept that is frequently used, particularly in a terrestrial environment, and not so much in a marine environment, because historically people thought that, well, things in the marine environment, they disperse so widely that this isn't going to be hardly useful because everything is everywhere. So it's a panmictic, everything is everywhere kind of a world in the environment. But for people working at patchy places like hydrothermal vents, this is potentially really attractive because individual vents are fragmented and they're discrete patches that can be separated by tens or hundreds of kilometers. And that the vent zones are then distributed, you saw in that previous map, into biogeographic regions around the world. So if they are a metapopulation, where sometimes a taxon can be missing from a place and then dispersal will allow the recolonization, that's quite encouraging, because the metapopulation effect is essentially not keeping all of your eggs in one basket. That if you've got a series of 5, so my fingertips are 5 different vents, and then every one of them, maybe 2 of them go extinct, we still have these 3 where the taxon exists, and dispersal from here can recolonize those 2, so on and so on and so on. So there's a protective metapopulation effect. Pardon my finger. And so let's just agree what we talk about when we're talking about dispersal, and it's not what Leslie Nielsen is encouraging you to do there, but it's this movement essentially from a birth site to where it reproduces. And so any movement, these are several different kind of descriptions, any movement that increases the mean distance, movement from one location to another, all of those count as dispersal at some place in the literature. We're going to focus on essentially realized dispersal, that first bullet, because we're going to measure it in this assignment or infer it using phylogeny, so it would be things that actually got someplace and reproduced, and we're looking at their the effect of that genetically years later. So you can imagine why it's important, and genetics is one way that you would measure it. Another way that you might do it is mark and recapture, but mark and recapture would work well if you're looking maybe within the same place. So short range dispersal at a particular habitat, maybe between adjacent, so with a regional dispersal within the same zone, but long range dispersal is notoriously hard to measure using direct methods, and so we often tend to use indirect methods like genetics. And one element of that's challenging in the marine environment that I've alluded to already is that unlike the terrestrial environment, where we have a bias towards thinking about, well, there's areas between these ponds that maybe the Daphnia can't live in, but they can kind of get there, or we can imagine that they could hook onto birds' legs, or they can get places through intervening areas where they can't. And the bias that we've had in the past in terms of thinking about not thinking about the importance of dispersal or patchy populations in the marine environment is because we thought most things were most places. And so we thought things just disperse and they go everywhere in the current. So let's revisit what we've already talked about this year in this course about marine larvae and about how marine larvae disperse. So we know that most adults at hydrothermal vents are sedentary. And so it would be natural to presume, or a natural first kind of assumption, that the connection between any one of these sites is not going to be by a wandering copepod, but by a drifting larva. And this ties us in to some of the discussion we've already had in class. which is about the importance of marine larval dispersal. And we've talked about that there's planktonic and non-planktonic larvae, and within the planktonic larva, there's a kind of a continuum between the planktotrophic larvae, long-lived and feeding larvae that tend to exist for a long period of time in the plankton, or lectotrophic larvae that get a yolk and they're short-lived and they don't need to feed. And there's a whole bunch of different kind of population traits that we talked about. In terms of lectotrophic, the non-feeding larvae, they have a higher lipid content, they have a larger egg, chemical defenses, and a positive buoyancy, whereas planktotrophic larvae are often assumed to remain in the water column longer than lectotrophic larvae, and they have a greater potential for dispersal or loss. And when meta-analyses are conducted upon taxa for which we have, or species for which we have data, that tends to be what we've seen. So this is a measure of egg size from a meta-analysis that was published several years ago for non-planktonic, lectotrophic, or planktotrophic. And it tends to support, the generalizations that I've made, but I think it influences the fact that this is a continuum. We're not talking like you can be only this or only this. So along this kind of the channel between England and France, this continuum we have from planktotrophic to lectotrophic, you see, some lectotrophic larvae are very, very small. Yes, but in general, there are more larger larvae that are lectotrophic, and the planktotrophic larvae, some can be a little bit big, but most are very, very small. And we talked in class about some of the advantages about being planktotrophic and existing in the water column for a long time, that you might be able to exploit temporary food source, you can colonize new habitats or recolonize habitats, expanding a range or repopulating a metapopulation in this case. It tends to, from a genetic perspective, we would measure it as a plus because it would avoid competition with SIBs and local competition with close relatives, because generally you kind of tend to go away from your parents and from your SIBs. And if there's habitat catastrophe, this gets you out of it. This is a get out of jail card because you're going to a new town. Now, the other element that has been talked about here, so there's egg size, but we keep talking about larval duration. So altering this strategy from lectotrophic to planktotrophic, altering the time that the larva spends in the current. And so again, now this time they've switched, so we've got non-planktonic here spending, and this is larval duration in hours. So this far end of the graph, this is like years. But the non-planktonic not spending much time at all, close to like generally zero in the plankton. The planktotrophic spending longer periods of time. Some spend a short degree, but then there are some that spend quite a long period of time, especially compared to the lectotrophic, where most of them spend a very short period of time, several hours. And so that longer time then results in potentially much greater distances. So the distances that can be traveled by a planktotrophic compared to a lectotrophic larva could be hundreds or thousands of kilometers over months and years instead of dozens or hundreds of kilometers over days or weeks. And you can see that modeled here, where across the X and the X labels up at the top, because we've got taxa labeled at the bottom. But here in days and the distance traveled, you can see thousands of kilometers. And then there's the shape of these curves that would be modeled. That's not really the point here of that particular paper, but that you could investigate those kinds of things based on different strategies, different larval strategies, potentially morphologies, and different short-term or shorter, smaller spatial scale experiments that let you kind of figure out this kind of dispersal kernel, it would be called. Now, genetically, how do we estimate dispersal? And this is clearly not a marine deep sea vent, but this is a tree. And so one of the ways that we would do it, we would use genetics to do it, would be using trees or phylogenies. Now, genetic inferences of dispersal, there's like decades and hundreds of years of interest in this topic, because with the very early noting that Darwin was part of, that gene flow so the consequence of dispersal, tends to homogenize variation at a place. Darwin observed that isolating populations was a major factor in promoting evolution. Think about his work with breeding populations, domesticated populations, domesticated birds, but also think about islands and the importance of islands to lots of him coming up with evolution via natural selection. And that's because increased population structuring should result from reduced connectivity being reduced gene flow. And there's many, many ways to estimate dispersal from genetic data. And if your career takes you in that direction, some of them I think are quite exciting and cool. But one way is via phylogeny. One of the simpler ways that we're going to do is to infer it based on the shape and the relationship of inter and intra-specific relationships visualized on a phylogeny. The little schematic here is just that we've got these isolated sites. Perhaps these are thermal vents that are more strongly connected by a planktotrophics, for instance, or weakly connected by something that's falling out of the current more quickly. So a reminder about what we're talking about when we're talking about phylogeny. So a couple of things. One of the things, some shared experiences that I want to make sure, shared starting point that I want to make sure we start from. When we talk about the branch length, we're talking about variation, and so understanding, calibrating that is important. So here we have a scale bar. Your phylogenies that you're going to make will have scale bars. So it's important to look at that and say, well, okay, so there it's 0.9% or roughly 1%, right? So this is a scale bar for a mitochondrial protein coding gene. And this is a barcode tree, so cytochrome C oxidase 1. And often what is inferred is that for those protein coding mitochondrial genes, when we look at isolation via percentage there, we plus or minus think about 1% or 2% per million years. That can vary a lot. But just to say that we're talking about we're inferring big changes via an extant distribution, via changes that could have taken hundreds of thousands to millions of years to add up. And another reminder that a tree like this can show you both inter and intra-specific differences. And let's talk about how to measure. So there's their scale bar. Let's say we wanted to measure two populations here, A and B. Now, just eyeballing that from the tree, one of the ways you could do that is say, well, if that scale bar is roughly 1, so then we've got 4, 6%, 8%, 11%. So population A and B are 11% divergent. And another way to think about this is that potentially we've got a break right, there's my annotate tool. Let's put a break right here and say this is a species and this is another species down here, okay? So A is a member of one species, and this population is, or this other cluster in the phylogeny is another population of this. So this is intra-specific variability here. Inter-specific, between species variability, would be that calculation between A and B. So you can see both, and you can measure and make inferences about both within species and between species processes here. Let me get rid of those things. All right. So here's the third layer, what are we up to? Fourth layer. So we've got deep sea thermal vents, and we've got the idea of patchy distributed deep sea thermal vents across the world and within different biogeographic regions, but isolated from each other and potentially within those regions, connected potentially by dispersal. So we've got deep sea thermal vents, a metapopulation approach, and dispersal, and the importance of dispersal, and then genetic estimates of how to understand how isolated one or the other is. Then, so that brings us, I guess, to 5, the fifth layer of the onion, which is the deep sea is no longer an isolated place that we can presume is not going to be influenced by our our development pressures. And so there's enormous interest in mining in the deep sea. And so we're going to talk about this, and I'll call attention to it as it goes along, but some of the data that I've got and some of the processes that I'll present, there's essentially mining at the deep sea that is in general, and then there's mining at thermal vents and mining for nodules. And some of the pressures, some of the consequences will be unique to one or the other, but they're all, I think it's worth considering all of them at the same time today. So this also goes back about 50 years, that around the same time Ballard and company were bobbing around and jigging with Angus on the bottom of the ocean, there were other ships involving other companies that were out there not looking to discover and document life, but looking to discover and document areas that they could mine, because this was seen as an enormous economic opportunity. And so this is this little video from Vox is about nodule mining, so outside of thermal vents, but also at the deep sea.
These discoveries sparked the interest of multinational companies in the 1960s and 70s, who were gearing up to mine the ocean floor for industrial uses like electrical wiring, stainless steel, fertilizer. Companies from China, Japan, the Soviet Union, Australia, the US, and European countries descended on the Clarion-Clipperton Zone. They tested mining equipment and took some of the first photos of the rocks on the seafloor. It was during this early chaos at sea that the Maltese delegate to the UN made a pivotal speech warning against repeating the mistakes of colonialism. He said that the race would reserve the plurality of the world's resources for the exclusive benefit of less than a handful of nations. The strong would get stronger, the rich, richer.
All right. So this is a picture of that gentleman that was just mentioned the last. So this is Arvid Pardo, who was writing about these motions and speeches that he was making in the UN at the time in the late 60s and early 70s, and was thinking very much outside the box. This was not conventional thinking to either to think about the potential, the richness of these elements concentrated into nodules or concentrated at hydrothermal vents on the deep ocean. The estimates of marine wealth were kind of staggering. And so what he was asking the delegates at the UN to do was to really engage in science fiction forward thinking to avoid errors that had colonial errors and short time frame thinking errors that we've made and continue to make terrestrially, where the consequences of the development are not thought about or evaluated by an independent body before granting licensing agreements to go out and to be able to mine. And this was successful, which was kind of amazing. And so in 1982, the UN met and updated the international law of the sea and created the ISA, or the International Seabed Authority, which would meet and could give out mining agreements or could sign licensing agreements as long as three conditions were made. That the company demonstrated that it would benefit not just one region, as particularly one region in the global north, but it would benefit all of humankind. That it explicitly considered where it was coming from as pertains to developing or nations. And thirdly, that it protected the marine environment. This would be great, right? This sounds great. Well, if you've been watching the news, you know that, and hearing about deep sea mining, you know that perhaps things might not be as great as all of that. So these are just a kind of a soupçon, a little amuse-bouche of different news items from the last year or so about fears that we have, that different biologists have around the world about this race, this increasing accelerated push to take advantage of these elements. The elements there's a real push to develop the elements because many of the elements are those involved in batteries. And so developing a global potential to move away from a carbon-based economy requires us to think about batteries, whether we're storing from the sun or storing from the wind, and those elements are often very destructively consumed from a terrestrial environment. And so the mining company would say, well, this is great. This concentrates mining into the deep ocean far, far away from any people, and it's not going to affect many things, and people will never see it. So the products are removed and brought to the surface in a number of the ways. For the nodules, and it's basically
To access the rocks like the ones lying,
easy, a nod like a Roomba, a big chunky Roomba that goes and zips all these things up, and you think, well, that looks pretty cool. And according to the and it looks pretty cool, and the bottom didn't seem to be damaged. And we know that mining in a terrestrial world has extensive, catastrophic environmental consequences. And maybe mining on the deep sea isn't that big of a deal. But the Roombas are like this. They're quite big. They're even bigger than that. So what kind of consequences are there to this kind of a disturbance to these areas that have been potentially undisturbed for millennia? Well, not to get into this here, but the consequences, the early the late 60s and the early 70s, when some of those first mining explorations were made, not with modern technology like this, but with the same kind of simple kind of like dredging. Those dredges are still visible. If those sites are revisited today, you can still see 50 years later the effect. There hasn't been a kind of a recolonization from whatever was living there. So remember this plot about the distribution of mollusk life at thermal vents around the world. Now, if we were to overlay onto that plot for those thermal vents, exclusive economic zones in a light blue, a marine protected area in dark blue, and the mining licenses in a darker blue, what you'll see is that these deep sea thermal vents don't generally fall into a protected area. They fall outside of that. They'd be very dependent upon a UN body like the Seabed Authority to interpreting the plans of a company and evaluating their long-term plans for recovery. And so much of that endemic diversity we're worried about. And so here we've got a low threat to a high threat, and you can see a lot of these areas, particularly here in the Indian Ocean, we're quite concerned about. We're even more concerned because in April of this year, there was an executive order that directed the Americans' National Oceanic and Atmospheric Administration, the NOAA, to fast-track any and all permits for companies to mine the ocean floor for American companies in both U.S. and international waters. So a race that was just accelerated. What's the Canadian government's position on that? It doesn't currently have a domestic legal framework that would allow us to permit seabed mining. And no more recently, but as recently as February in 2023, the federal government confirmed that it wouldn't authorize seabed mining in areas under Canadian jurisdiction until such time as a rigorous, comprehensive blah, 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 blah. And in areas outside of our national jurisdiction, Canada continues to uphold the United Nations Convention of the Law on the Sea and the Seabed Authority. So potentially that's good news for our role in the international community, yes? So governmentally, perhaps, but there are Canadian companies that are real accelerants of this race to get and gather minerals from the bottom. One of them in particular is called the Metals Company, based in Vancouver, and is always amongst the first to apply. And in this case, following the April 2025 declaration of the American government, it was the first to apply to mine the seafloor. And doing this, particularly in the way of that executive decree by the Americans, is a way that bypasses the International Deep Sea Waters Authority, the ISA, which meant that it needed to benefit humankind, it needed to be directed towards less developed nations, and it needed to maintain the integrity of the biodiversity, the biology of that marine environment. So that's bypassed here by this process. So this is a really big company that's actually in the news a lot. This is a picture of their the co-founder and CEO. And if you want to later on, I'm not going to start this video here, but there was a whole episode last year in the summer, in the early summer, on Last Week Tonight with John Oliver about deep sea mining, and it features some of the Canadian Metals Company in particular. You can click on that later on, or you can scan down here and jump over and open up another window to watch it right now. Now, a couple of elements. So that's a Metals Company vessel up at the top. One of the elements, one of the elements of their exploration of these zones, one of the way they have collaborators at academic institutions, including the Natural History Museum in the United Kingdom. This giant, if you go to London, go to the Natural History Museum. It's incredible. And so that data, some of it is publicly available. And we know that more than up to 80% of the species that they have collected in their exploration zones are unknown to science. Now remember, the Metals Company is exploring those for nodules, right? They're not so much looking at this is a low estimate of novelty, of unknown. That if there's 80% on those flats where the nodules are growing, the endemicity and the proportion of taxa that are unknown to science goes only up into the 95th, 99th percentile. So on the flats, 80%, at the deep sea vents, 90 higher, 95 higher. But does that matter? Well, there's some statements from companies like, and I keep thinking about this Canadian one. This is Michael Clark, the environmental manager for the Metals Company, who in an article that you can read there, and the Narwhal talks about the fact that these 3 points. We don't need to know the name of every species that's out there. We don't need to know the distribution of every species. What we need to know is the magnitude of the impacts that we're creating. So I read that, and I have a bit of a problem with the logic stream, because to know 3, I would argue that you need to know 1 or 2. And what we're going to work on together in this assignment is a biological way of inferring isolation or connectivity, which lets us infer impacts of mining. And we can do that because most of these taxa are new, most of them are barcoded. And as they are described, as those barcodes are created, they're added to public data repositories. And you're going to use some of these public DNA barcodes to estimate the degree of connectivity in a small case, not for a whole range of taxa and not at all sorts of vents, but at 2 fields, several vents in one, several vents in another, and for 2 species of copepod. And I chose copepods because they're one of the most abundant taxa at thermal vents. There's 80 or more described species. There are about 80 species described, but there's many more that remain to be described. And because they have been barcoded, we can infer the effect of dispersal by making these phylogenies to ask how isolated is each vent, and then can their larval strategy be inferred using these publicly available DNA barcodes, and what consequences for those species will be associated with mining. Now, that's kind of the context. That's 40 minutes of context. There's another 30 minutes or so right down here where the example, a parallel example with a terrestrial invertebrate I go through, and we figure out how to show you how to use R. And I know a lot of you know how to use R. You've used it in different classes, but I also know that a lot of the times your use of it is you're done in isolation and perhaps isn't that positive. So my hope is that by doing it in this way together, that it becomes a more positive experience for you, and one where you can save this routine and you can imagine using it in courses, maybe other courses this year, but courses into your 4th year, into your honors thesis, into your graduate work if you want to do it. Finally, I have some suggestions about, that there's all of this work that's going into this project, and it's spread all over. There's 2 of them over the term. You've got a month to do this. The evaluation that we're going to do we've specifically designed to be a very brief one. It's 3 minutes of you talking about the results. And the results are a PDF that the R routine will create for you, 3 pages. And the video that I want is you kind of standing with a computer as if you were accosting somebody, chatting with somebody, in an elevator. And you had 3 minutes, it's a tall building, to tell them what you found, why that's important. Okay? And so there's some specific questions that are laid out within the R environment as you're doing the experiment. But these are my suggestions about how to do that. The rubric for this assignment is or the requirements for the assignment is that you're not reading. We don't want to see you reading. We don't want to see a prepared script. But that isn't to say that you shouldn't practice. So set your phone up or set up your Zoom window or whatever and record it. And talk about it. Then watch it back and stop it at 3 minutes. Watch it back and go, oh, that's not so good. I go, okay, so I didn't say this. I should have said that. I did say this and I didn't mean to. Watch it again, critique it, and redo it. Maybe pair up with a friend. This is one of my suggestions for your case studies every week. There's real value to doing these things together and not working in isolation. And then it gives you somebody to imagine. Maybe it's not you just imagining that you're talking to the camera. It's you talking to your friend who's kind of standing on the other side or holding the camera. Draw, annotate on the paper. That's a real powerful thing. If you've got a clipboard, so you're going to have a piece of paper, but put your paper on a clipboard, on a cereal box, on something, tape it to something so that you can gesture to it so that the TA who's evaluating these can see what you're pointing at and what conclusions, what inferences you're making. So this is the beginning. This is kind of the midway point. You've got the R example to look at, but my hope is that you keep this routine and that it's useful to you here. And that you might be able to adapt it and use it for other courses and assignments. And that, as they say, is it.
Oh, the lights.